So I just want to welcome everybody to the 2022 PKD-RRC Annual Symposium. Our mission is to develop and share resources and reagents and expertise to advance research in the field of polycystic kidney disease. I'm just going to give a brief introduction to our consortium. I think a lot of people have heard about this at various meetings, but I encourage you to visit our website. I'm just showing you some of the face pages here from all of the different reagents that we have available. So please, we have all types of in vivo models and reagents. We have antibodies and vectors, cell models, and clinical resources and biomaterials. Our website makes ordering easy, so please visit us. And I also want to encourage everyone to get involved in other PKD-RRC activities. We have a pilot project program. We have an annual symposium, which you are all participating in today. We have a monthly science roundtable where we invite speakers to discuss their research. So you can sign up for to get notifications about that on our website. We are promoting community engagement. You can follow us on Twitter. Sign up for our mailing list, and we also have a discussion board where you can bring up any scientific questions you have to start a discussion with people in the community. And like I said, this is all available on our website. So just to summarize briefly the objectives for today's symposium, we are hoping to identify state-of-the-art techniques that could be applied to advance our understanding of PKD biology. We want to continue to discuss the challenges and gaps in PKD research that if we could address them, we would be able to move the field forward. We want to promote broad awareness of advances in PKD research and highlight existing PKD research resources. As always, we want to foster interaction among members of the scientific community. We want to, most importantly, obtain feedback from you guys in the community with respect to what tools and resources we can develop to advance PKD research. So I'm just going to give you a brief overview of the first day, which we're about to start. We're going to start off with a session on novel aspects of polycystin function and signaling. We're going to have a session on advances in pediatric PKD. We will break for lunch. And then after that, we're going to have a presentation of trainee abstracts. I just want to point out that all of those are available on the symposium website. So we've gotten a lot of submissions. We've selected 5 to be presented. After that, we'll talk about new technologies, basically omics, followed by a panel discussion. And then we're going to separate out into breakout rooms where we really want to elicit community feedback about what your critical needs are. So just a few housekeeping issues. Everybody is going to be muted and there'll be no cameras except for the speakers and the session chairs and panelists. Okay, we're going to handle, because of the number of people that we have registered, which is well over 200, all questions are going to be handled through the chat. So when you put your question in the chat, please submit those questions and comments to everyone. For speakers, we're going to give you a 3-minute warning when your time will be up. If for whatever reason, if you're a speaker, session chair, whatever, in the audience, if you have a technical issue, please contact John Hare or Stacy Turner through the chat. Very importantly, we're going to be distributing an evaluation survey a couple of days after the symposium, and please be sure to answer and return that so that we can take into account all of your feedback for next year's symposium. On day 2, which is a half a day of talks, after that, there's going to be a session for trainees, okay? If you didn't sign up and you're interested, please email Stacy Turner at bridges@som.umaryland.edu, or you can do that through the website and we'll make sure to get you the link. So then I would just want to say thank you to everybody who's participating. I know we've had a lot of PKD symposia. We had the FASEB over the summer. But I do want to thank you all. I want to thank all of the speakers, the moderators, the session chairs, and panelists. This symposium was put together by a wonderful organizing committee listed there that worked very hard to try to make sure that we didn't overlap too much with the FASEB. And we also had 3 trainees who helped to put together, I think, a really wonderful trainee session for day 2. And then finally, I just want to thank NIDDK, who supported the PKD RRC and this symposium. So with that, I think we can go to the first session, which is novel aspects of polycystin function and signaling. And the session chairs are Marcus Delling, associate professor at the University of California, San Francisco, and Francisco Altamirano. So with that, I'll turn it over to them.
All right. Good morning, everyone. Thanks, Terry, and the organizers for putting together this fantastic line of speakers. And yeah, it's a pleasure to kick this meeting off. And the first speaker of this session is Paul DeCaen from the University of Chicago. For those of you though who do not know Paul, which I believe are very few. So Paul is an ion channel biologist that has been working on ion channels for most of his research life. He received his Ph.D. at the University of Washington and did his postdoctoral training in the lab of David Clapham, and he now has his own lab at Northwestern University. So, and he will be talking to us about the molecular regulations of the polycystin complexes. Paul, you want to start?
Oh, you're still muted and your slides aren't in presentation mode.
Got it. Let me try doing something here. Apologies. Sorry about that. Do you mind if we just do it like this?
Yeah, that's fine.
Apologies. So thanks a lot for having me, and I appreciate the invitation. Wish I could see you all in person. I'm going to be talking about the molecular regulation of polycystin complexes. Obviously, we're here because of autosomal dominant polycystic kidney disease for the adult version and the recessive version found in children or in development. For AD-PKD, it's one of the most common monogenetic disorders. Affects about 5 million people with midlife onset. With the midlife onset, it presents opportunity for intervention. There is currently no drug cure. So we have some therapies, but they do not address the underlying cause. And they don't really delay cyst formation, rather delay the amount of time required for transplant. There's a clear established genetic component, and so this is where the vast majority, 95% of the cases, are caused by variants in 2 ion channel subunit genes, PKD1 and PKD2. Those are the gene names. They're also called in their protein form, polycystin-1 and polycystin-2. The majority of variants which cause AD-PKD affect PKD1 or the protein polycystin-1. For the remainder of the talk, I'm going to be just talking about them in their gene name forms, so I'll be using the PKD nomenclature. So polycystins are extremely cool proteins in the ion channel world. The PKD2 related subunits, which include PKD2L1 and PKD2L2, encode 6 transmembrane spanning proteins. These are canonical trip channels. But as we've learned through functional evidence, they seem to be voltage dependent and calcium modulated ion channel subunits. And we know that they can come together and form ion channels on their own. They can form homomeric channels. The ones that are related to PKD1 are 11 transmembrane spanning proteins, and they can heteromerize with members of the PKD2 type. So we have 2 different cryo-EM structures of heteromers now that include PKD1 and PKD2 coming together and form a heteromeric channel, as well as 1L3 and PKD2L1, forming what is thought to be a putative acid sensitive ion channel. And we know that, these proteins, they have are 11 transmembrane spanning proteins, and they have an ion channel homology region, which is in the last 6 transmembrane segments. So those ones are the ones that form those contacts with the ion channels of PKD2 and PKD2L1. And we also know that these have a GPCR or adhesion GPCR type of homology to the first 5 transmembrane segments. And so with this class of having this relationship with adhesion GPCRs, we know that they appear to be activated by cleavage at the GPS site, and that upon this cleavage, it either frees the stalk domain, which is located above the first S1 transmembrane segment, or perhaps the ectodomain itself actually functions as a ligand for the channel complex. So we have 2 channels, sets of channels, one that appears to be constitutively active in response to voltage and calcium. The other one appears to be autoproteolytically activated or ligand operated. So these advances are quite remarkable, and really kind of has led us to some really big breakthroughs in ADPKD. And the first structure was solved by Peter Shen, Yao Yang, and Erhu Kao. And they solved these structures at 3 angstrom resolution in lipid nanodiscs. So these PKD2 type channels come together, form a homotetramer that is 4 copies of the channel subunit, and they have a strong electronegative ionic pathway or selectivity filter at the top of the channel. Those are those red residues. And the selectivity appears to be preferring monovalence, but also conducting calcium. So they equally prefer potassium and sodium, but can also permeate calcium. And whereas comparing that now with the heteromeric versions of PKD2 associated with PKD1, so there is an electropositive ion conducting pathway contributed by a group of lysines that are aligning the S11. And this could be forming a number of functions that aren't completely understood. It could be either serving a function to create an electropositive barrier, such that if it's moved out of place during gating, perhaps through its ligand or proteolytic regulation, that these positive charges move out of the way and allow for the ion to move through. Or it could be that these positive charges are actually serving a function in selectivity, like bringing in water, such that either the ion of choice, whether it's calcium, sodium, or potassium, can become rehydrated upon entry into the pore. So beyond the kidney, I just want to give some information that it's, these polycystins can form different heteromers, and those things can localize to different places in the body. And so we have some stories that are starting to emerge as far as various polycystins that are involved in human reproduction and perhaps development. So beyond the kidney, where we have PKD2 functioning in these cells, we have, for example, PKD, REJ, PKD2, perhaps functioning as an ion channel involved in the acrosome reaction that allows for fertilization to carry out. And it's thought to be reacting with zona pellucida proteins on the unfertilized egg, as well as in our development. So such that in the embryonic node, we have both an organelle, which is has motile and primary cilia, which are thought to create a gradient of some kind of signal, which allows for asymmetric expression of the hedgehog pathway. So this signal has been debated whether or not it's nodal flow or perhaps a morphogen, which is moving from one side to the other, might set off a type of gradient that would allow for the or stimulus, which would allow for the activation of PKD2L1, PKD1L1, and PKD2L1 heteromeric channels in the primary cilia of crown cells. So one thing that you've noticed from this slide is already you see that we have ion channels which are not really found in the obvious places like plasma membrane. We see that already polycystins seem to be subcellularly localizing to unique organelles such as the primary cilia and the acrosome. And so as it relates to PKD research, we study polycystins in the primary cilia. So these are hair-like structures on the apical side of cells, and you can find them in a lot of different tissues. So whether you look at the brain hippocampus or perhaps the kidney collecting duct, like that we're mostly interested in, you can find this between 1 to 10 micron long structure. And it's less than a half micron in diameter, and they house these unique polycystin ion channels. So the methodologies to study these ion channels in primary cilia have been contributed by a lot of people, and that includes Nancy and Steve Cleaney at the University of Cincinnati, David Clapham, your host, Marcus Delling, Takanori Inoue, and others. So our lab has been able to generate small electrodes to actually record these ion channel currents directly from the plasma membrane. So we find they're about you typically can get these down to about 23 mega ohms in resistance, so less than a micron in diameter. And with that electrode, we can then seal onto the plasma membrane and record currents. In doing so, you can also use these in conjunction with different calcium sensors. So in this case, we're using an ARL redshifted gecko. There's different varieties that are out there. And in conjunction with doing electrophysiology, you can actually record changes that are happening in this primary cilia in response to activation of these ion channels, which are those stochastic opening events shown there in the green traces. And we could use super resolution to actually image the trafficking of polycystin 2 in this case, localizing to the primary cilia. So polycystin 2 is tagged with GFP, and we can see these small little vesicles that are then docking into the primary cilia, which has been co-labeled with ARL in red. And so using these approaches, we've been able to determine a few things. One, that the cilium has a high resting calcium concentration. It has a more depolarized resting membrane potential. And by moving through different parts of the different ciliated tissues, we've been able to find that different polycystins actually localize and function in diverse tissues. Sorry, I'm making everyone dizzy. This is just a bit about super resolution. So it's really important to try to visualize primary cilia using super resolution, because using 3D sim, you can actually suss out the depth of your signal, which can be, confused with signal that comes from the base of the cell. So we have two images here that of a cilia. One's a confocal, one's a 3D sim. And you can see that the confocal exaggerates the size of the cilium, as well as it doesn't really allow you to visualize the depth of where your signal is coming from. So with that, we had about 20, several questions. We consider them 20-year-old questions. So that despite our strong understanding of the genetics in AD-PKD, we still don't understand a lot of the structural features and physiological stimuli which are responsible for opening polycystins. We don't know how the disease-causing variants impact their function. And we, of course, everybody would love to get to understanding the polycystin pharmacophore and whether or not we can, leverage that for AD-PKD treatment. So at the core of PKD2 is a voltage-gated ion channel. So if we took apart this channel and just looked at a single subunit, you can see that in blue you have a top domain, and in the green you have a pore-forming domain. These are set up by the S5 and 6 segments, as well as a voltage sensor domain, which is then has two gating charges, and these positively charged residues that are shown there in purple are able to sense the change in membrane potential as the cell depolarizes. And of course, we have a C-terminal domain, which is still unresolved. So if we just look at the core and ignore the top and the C-terminus, you can see that this channel, PKD2, is very reminiscent of other voltage-gated ion channels. This is another sodium channel that we participated in structuring. So this is the subunit of it. We have, in this case, 4 positively charged residues, which sense the change in membrane potential, pore-forming regions in the S5 and 6. And it is found in the open state. And you can see that R3, or charge 3 and 4, which are conserved with PKD2, have transferred out about 1 helical turn. And other evidence for this channel's voltage sensitivity comes from the fact that if you compare it to PKD2 to the 2L1 channel, which has then been captured in the open state, you can see that this voltage sensor is found slid up or in the activated state, and the pore-forming region is splayed open to allow for the ion conductance. So some of the polycystin, so we propose that the polycystin gating charges actually sense the electric field, forming state-dependent interactions prior to channel opening. So this is a Rosetta-based animation of how that voltage sensor works. So as the membrane depolarizes, you can see that the positive charges move up. And then ultimately in its quaternary state, the voltage sensors then, that state dependence allows for the opening of the ion conducting pathway. And a lot of the interactions which are responsible for how this voltage sensor then opens the gate through perhaps the S4, S5 linker, or through the top domain have still remain to be resolved at this point. But we know that we can measure those transfer of gating charges directly by first of all, neutralizing the ion conducting pathway, either through mutation or perhaps pore block through gadolinium. And what you're left there with are these small capacitive type currents, which are dependent on expression of that channel. And this gating current can actually then be transposed on a voltage charge continuum and then plotted relative to the tail current. And we can see that tail current, which is coming from the ion conductance through the central pore, is actually occurring on more depolarized potential, suggesting that actually the gating charges have to move first, both over time, as you can see here in the kinetic of the depolarization of, or the activation of the gating current relative to the central pore current, and as far as on the continuum of voltage. So it has to occur earlier relative to the conductance of the pore. But some of the questions we had, well, given the positive membrane potential, what's the physiological relevance? So we know that calcium actually functions as a bimodal regulator of PKD2. So it, one, allows for the shift in membrane, the sensitivity of the channel to open. So in this case, we're recording cilia with 10 micromolar calcium inside the pipette. And we are, and then comparing that to 100-fold less, which is what you would typically find in the cell body, we can see that the voltage dependence is much more shifted to the positive direction, suggesting that the open probability is then being enhanced by the increase in intracellular calcium. We can do recordings where we're measuring the calcium fluorescence in relative to depolarizing the cell with high concentrations of potassium. So in this case, what we see is that in response to high K, we have stimulation in the single channel currents. And that is also occurring at the same time as calcium is being recorded. We can see that it doesn't, once a depolarization happens, both the channel and the relative amount of calcium actually mirror each other. So it suggests that actually the stimulation of calcium as it comes through here is actually turning off or desensitizing the calcium the ion channel conductance. And so we kind of think of these things as two different switches. Calcium dependent modulation is kind of an on switch for opening the channel. And if it's left on at a high level over time, it kind of shuts off or desensitizes the channel. So many of the things we're trying to do is try to figure out what are the calcium responsive elements responsible for this, either the modulation or the desensitization effect.
3 minutes.
So some of the structural features and physiological stimuli responsible for opening PKDs we've gone through, how disease-causing variants of impact the channel's function is something that we are working on as well. So we've set our sights on the top domain and how it regulates the channel. And the reason why we are focused on it is because the top domain is the site of most of the AD-PKD variants which affect PKD2. So we focused on 3 sites within the top domain called the finger 1 motif. And so there are redundant sites where several missense variants have been reported in the literature and through the Mayo Clinic's database. And these sites, based on the molecular structures from ERHU, are involved in disulfide bond formation. Another one is involved in hydrogen bond network, as well as a pi-cation interaction. And all these are occurring within the top domain itself. So we tested all those mutations, and we had 2 hypotheses as we were setting out. Variants will alter channels assembly or disrupt the cilia localization. That could be one way in which you can get a loss of function, or the variants will disrupt the channel's function directly. So ERHU solved the structure of this of the wild type channel along with a disease-causing variant. We found that the channels do, in fact, still associate and form oligomeric structures. And in fact, they still localize to the primary cilia, suggesting that, okay, these variants are not disrupting the native localization of the channel. But when we measure the channel directly from the primary cilia, we find that the voltage dependencies shift to the positive direction, suggesting that the nature of the loss of function for these AD-PKD causing variants is actually a voltage dependent shift. That was really interesting to us because given that these things are still available in the channel, in the cilium, that they can perhaps be still drugged. As well as these are interesting because it suggests that this is a new motif that's involved in actually controlling the gating of the channel, which was really a surprise to us. But perhaps it shouldn't have been, because there is already precedent for other bimolecular reactions, such as the form of protoxin 2 associating with voltage-gated sodium channels, which then shift the voltage dependence of these channels, which may be analogous to the way that the top domain is modulating PKD2. So in conclusion, elevated ciliary calcium increases the open probability of PKD2. It's followed by desensitization. And this likely does not involve the EF hand based on published work, which I did not have a chance to show you today. Disease-causing mutations in the top domain always alter PKD2 or polycystin 2's channel function through impaired channel gating. And of course, variants in PKD2 still allow for the channel to localize to the primary cilia, suggest there might be a druggable target. Perhaps we could develop modulators that can shift the voltage dependence back to the native state. And with that, I'd like to thank my collaborators and those in the lab who did the work: Leo, Genesis, Tui, Shou, Zhao, and the Megan Larimore, and Megan McCollum, as well as my collaborators, Erhu, Andy, Peter, Yao Yang, and Steve Somolo, Peter Harris, and Vladimir Yarovoy. And of course, my funding. And thank you for listening.
Thank you very much, Paul, and thank you for staying perfectly on time. So if you guys have any questions, as Terry said earlier, please type them in the chat, and I will read them out loud, or Paul is probably also reading them. Yeah, and so maybe I can start with the first question. So Paul, you can show very nicely that kind of membrane depolarization primes the polycystin channel for activation. So do you want to speculate what initiates this membrane depolarization in the cilium? I mean, given that we don't, I mean, do you think there are other channels that prime the polycystin activation, or is this the plasma membrane depolarization that depolarizes the ciliary membrane? What are your thoughts here?
I think it's an open question and a great question. So like maybe it's perfectly conceivable that depolarizations, as you know, can affect the primary cilia. So you know from your work that effects in calcium from the cell body can easily cause an influx of calcium to the cilium, but probably not vice versa. So it seems to me that whether you're finding a polycystin in a neuron or perhaps in a collecting duct, effects that are happening at the plasma membrane in the form of either calcium transients or even voltage can then affect the primary cilia and polycystin channels and open them. But that doesn't mean that that's the only way, right? So there could be, these are the voltage sensitive ones, perhaps the heteromers are actually involved in something more special. And certainly the heteromers, and since the disease causing variants seem to be distributed more asymmetrically towards PKD1, perhaps actually these are the more physiological consequence as far as ADPKD. So there could be, for example, in a ligand or perhaps audio proteolytic effects that are happening within the cilium, which are gating those specialized forms of polycystins. So I think it's an open question and a great question.
Okay, so there's another question here. Is there a HTP way to measure PC activity that would be amenable to drug screening?
Yeah, I think it's a great question. So the problem is I don't know what technology is out there. We've been dabbling a little bit in this, but you're going to need to have a primary screen for looking at calcium fluorescence in primary cilia, which has really high content. And what I mean by high content, as far as a buzzword, is one, being able to handle the throughput of a lot of compounds to screen, and second, being able to have the resolution over time and within space to see these effects occurring in the cilium. So I imagine that should we develop such technology, that it's going to need to be very faithful as far, you're going to have to screen and weed against effects that are coming from the cell body, because you can easily imagine a lot of false positives where you're going to get calcium transients emitting from the cell body. But should a high content screen be developed, perhaps using calcium fluorescence, that would be fantastic.
Yeah, I couldn't agree more. So yeah, are there any more questions? Otherwise, I think it's 7:25. Paul, thank you very much. I think if there are more questions, just type them in the chat or email Paul directly. And Francisco, do you want to take over from here?
Yeah, thank you so much. Good morning. So the next talk is by Dr. Demet Araç. So she was an undergraduate in University of Turkey. Then she moved to UT Southwestern, and she elucidated the mechanism of neurotransmitter release. And then she did the postdoc in Stanford University to study the structure and function of cell adhesion proteins. And in 2013, she started her own lab at University of Chicago. So she's an associate professor there, and she will present lessons from the adhesion GPCR field for PKD. Thank you so much.
Hello. I'm Demet Araç, and first I want to thank Terry and other organizers for this opportunity to give a talk in a polycystic kidney disease meeting. Actually, giving such a talk has always been my dream, I would say, for the last 10 years or so. The reason is that I actually do work on adhesion GPCRs, and very briefly worked on PKDs in the past. But what we have learned from working on adhesion GPCRs, we have found out that there are similarities between adhesion GPCRs and PKD1. That's why I believe that what we learn from adhesion GPCRs will be useful for the PKD field. And in this talk, I will try to tell you about adhesion GPCRs, but you can think, whenever I am talking about adhesion GPCRs, you can think that this also hopefully applies to PKDs. So, polycystic kidney disease 1, PKD1, is a cell surface receptor. And cell surface, it has 11 transmembrane helices, as you all know, and it has this extremely large extracellular region. Cell surface receptors are so that they do receive signals from outside and pass that signal to inside. So based on what I have heard from the first talk, I would guess that they are receiving the signal from outside. Either it can be a different ligand, a protein ligand of small molecule that might be even interacting with the transmembrane region, or it is possible that they are sensing a mechanical force from outside, maybe blood stream or anything else. And I don't know the PKD field very well. I know briefly, and yesterday I looked around some of the literature, and I was happy to see that in the last 10 years, actually there has been the structures of PKDs and PKD2s that has been resolved. And I found this paper, basically the transmembrane region of PKD1 has been solved in complex with PKD2. And today I learned that there are even more structures that are available. But what this paper said is that the 3 PKD2s and 1 PKD1 form a complex. And they designate this last helices of PKD1 as the channel, and the first 5 transmembrane helices of PKD1 as the GPCR. So basically, it looks like PKD1 is a combination of some kind of a GPCR and some kind of a partial channel that contributes and makes a full channel maybe together with the PKD2. So based on this, now I'm going to focus on the GPCR part. And one other thing I wanted to say is when we look at the pharmaceutically available drugs that are in the pharmacies, about 50% of currently marketed drugs work either by targeting a G protein coupled receptor, a receptor on the cell surface, or an ion channel. So it looks like targeting PKD 1 especially, and PKD2, is a really good, there is hope, I believe. So what do G protein coupled receptors do? This is a typical GPCR. It has a 7 transmembrane helix, and it binds to things that come from outside. These can be neurotransmitters, small molecules, or large proteins. And what they do is couple this signal to downstream intracellular signaling pathways by coupling to trimeric G proteins. But in the case of PKDs, I think that the signal passes to the PKD2 probably, and tells PKD2 to work as a channel or not, to open or not. So this is a typical GPCR. Adhesion GPCRs is a subfamily of GPCRs. There are about 1,000 GPCRs, about 33 of them are adhesion GPCRs. And the adhesion GPCRs have been classified in 2003. They are the second largest GPCR family. There are 32 members in humans. They are very large and complex genes, and they are the least studied and understood family of GPCRs. So adhesion GPCRs are involved in many different diseases. They have the 7 transmembrane helices like other GPCRs, but they have this very large extracellular regions, not as large as PKD1, but still up to 2,000, 3,000 amino acids. There is this famous GPCR proteolysis site where the protein cleaves itself. The autoproteolysis happens in the endoplasmic reticulum when the protein is being folded. So in the adhesion GPCR field, about 10 years ago, until about 10 years ago, it was believed that this region, before the GPS cleavage site, this region was a stalk, and that it is not very important. It is not conserved and not functional. But during my studies, when I was a postdoc in Axel Brunger's lab, we realized that this region, together with the GPS motif, actually makes up a structured, folded domain. And we named it the GPCR autoproteolysis inducing domain, the GAIN domain. So it turned out that all of the adhesion GPCRs, all 33 of them, have a GAIN domain. They have other domains also that are different between different adhesion GPCRs. These other domains are variable between different adhesion GPCRs, but the GAIN domain is always there, never anywhere away from the transmembrane region, always close to the transmembrane region, about 6-7 amino acids close to the transmembrane region, and it is always there. So the core of an adhesion GPCR we identified is that there's a GAIN domain and there is the 7 transmembrane helices. So basically we said that the GPS motif is not an autonomously folded domain, but it is only a small part of this much larger novel GAIN domain. And we determined the crystal structures of different GAIN domains. So this is the GAIN domain fold. This is where the cleavage occurs. This last orange strand is the cleaved peptide that is left after cleavage. But one thing is that autoproteolysis, cleavage of the protein, does not cause dissociation of the N-terminal fragment from the C-terminal fragment. So cleavage occurs, but there is no separation of the two parts. And this orange peptide that you see here, it is not accessible from outside. It is hidden within the GAIN domain. When you fill all the atoms in the GAIN domain and look from outside, you are actually unable to see this orange peptide because it is hidden inside the GAIN domain. So all of these, I believe, is true for PKD as well. So this identification of this domain, before that, and what we have done is we searched for structural features of this domain. Basically, there is the alpha helices and beta strands that are in this domain, and which this was actually a novel fold, a novel domain, they all, and we found that the secondary structure exists in all of the adhesion GPCRs. It exists in very early organisms, including slime molds, which are the single-celled organisms that have transitioned to multicellular animals, Tetrahymena, C. elegans, and Drosophila. And by the way, PKD, I believe, also does exist in C. elegans. So they all go to very early organisms. And this cleavage site, when we look at these very early organisms, does exist in very early organisms as well. So autoproteolysis seems to be evolutionarily conserved. So basically, I said it all exists in all 33 adhesion GPCRs. But then we looked at polycystic kidney disease sequences, and we looked at the 5 human PKD1, not PKD2, but PKD1 like 1, like 2, like 3. All of them do have the gain domain. And all of them, I believe, I can't remember now, has the autoproteolysis site in there. So basically, in summary, PKD1 also has a gain domain, which is so extremely conserved, evolutionarily conserved in other organisms, in early organisms, and in adhesion GPCRs. And this is important because let me tell you that this is the autoproteolysis showing the PKDs, the important cleavage site, and the presence of the gain domain. And that when we looked at the PKD disease mutations, the gain domain of PKD1 looked like it was a hot spot for PKD mutations. And basically, the reason why this is important is because gain domain, since has proven itself to be an important domain for the activation mechanism of adhesion GPCRs. And I believe that this might be true for PKDs as well. So basically, how adhesion GPCRs are activated. There's lots of research that has been done since the identification of the gain domain. Basically, this is the gain domain. This is that green cleaved peptide that upon cleavage, it stays together inside of the gain domain. Probably by binding of a ligand that binds to the extracellular region of adhesion GPCRs, or by mechanical force that might be acting on the outside of the cell. It might be blood flow, it might be another cell, another ligand on a cell binding and pulling on it. I don't really know what it would be for PKDs, but it is suggested that ligand binding causes shedding of the extracellular region from the rest of the protein, so that this peptide, which is called the Stahl peptide, is now released. It's free, it is outside. And it has been shown for adhesion GPCRs that this peptide binds to the 7th transmembrane helix of the adhesion GPCR. Now there are structures of this tetrad agonist bound to the transmembrane region, and it acts as an agonist to activate the GPCR. And what does activation for a GPCR means? It means that it couples the trimeric G proteins and sends the signal to inside of the cell. In the case of PKD, it will be different. I don't know what it will be, but it might be still be coupled to G proteins. I don't know, but it is possible that tetrad agonist, this peptide, can be acting on the G protein part of the GPCR part of the PKD and activating the receptor. So in the adhesion GPCR field, this mechanism was widely accepted. However, there are some problems with this mechanism. So not all functions of adhesion GPCRs depend on cleavage. Some adhesion GPCRs are not cleaved. So how are these receptors activated? And there are some physiological functions of some adhesion GPCRs that can be rescued by a wild type receptor, but also receptor that is mutated, that is autoproteolysis null. So even if the receptor cannot be cleaved, it can rescue some of the phenotypes of adhesion GPCRs. So this mechanism cannot explain everything. In addition, every time GPCRs are used again and again, the ligand comes, binds, goes away, the GPCR is turned off, and then ligand comes again, on and off. So if this mechanism is true for every case, then this is going to be very costly for the cell, because every time this is an irreversible mechanism. Every time the cell has to make a full-length receptor again and again so that it can be activated. For this reason, there is a need for a second mechanism. Not that the first mechanism is wrong or anything. We think that multiple mechanisms work to activate adhesion GPCRs. This may depend on the receptor. This may also depend on where the receptor is functional, because there are multiple functions for a single adhesion GPCR. So this second mechanism, we think, is Stachel peptide independent modulation. So basically, in this mechanism, the extracellular region is close to the transmembrane region. It might be doing some transient interactions with it. And these interactions, existing or not, by binding of a ligand, again, by activation, by mechanical force maybe, that is coming from outside of the cell, it might change these minimal interactions between the extracellular region and the transmembrane region. And as a result, this might modulate the receptor function. This explains how uncleaved proteins can be modulated. This explains how even autoproteolysis null proteins can rescue phenotypes. This also is a reversible reaction. The ligand can come off, the receptor can be turned off and on again. So basically, I am not going to go into the details, but this brought us to the question of what is the role of extracellular region in adhesion GPCR function. And over the last 10 years or so, my lab has been working on many different adhesion GPCRs, their extracellular regions, their ligands, what binds to them, what binding of ligands, how does it affect the downstream signaling, and how can we modulate this? So modulation of adhesion GPCRs it is possible by binding of synthetic proteins. These are antibodies. 3 minutes. Synthetic proteins that we can engineer to bind to the extracellular regions, or it can be antibodies that bind to specific regions of wherever we want, basically. And binding to the extracellular region can change the downstream intracellular signaling. And by that, I mean cyclic AMP levels, calcium levels, whatever G proteins, GPCRs coupled to. So this is one way of activating adhesion GPCRs. And another way of activating general GPCRs or modulating their function is by small molecules. Basically, almost 30–40% of drugs are small molecules that target G protein coupled receptors. And what they do is the small molecule binds to the transmembrane region, to the orthosteric site in the GPCR, and changes, most of the time inhibit the overactive GPCR. So this is the typical way of modulating a typical GPCR. But what I am saying, what we have said specifically for adhesion GPCRs is that because adhesion GPCRs have large extracellular regions and they do affect downstream signaling, that is why it is possible to engineer proteins that bind to the extracellular regions and change their function downstream signaling by targeting the extracellular regions as well. So, okay, basically I will skip this part where we did some more studies to show the presence of the second mechanism as well as the first mechanism. I will skip all the way to the end. Basically, in the adhesion GPCR field, we can come to the conclusion that the extracellular region of adhesion GPCRs is critical for downstream signaling. Still, we do not know everything about adhesion GPCRs. There is so much we do not know. But one thing we definitely don't know is how does the extracellular region communicate with the transmembrane region? And we can say that both the extracellular region and the transmembrane region of adhesion GPCRs can be targeted for drug design. And I said we can think of everything I say about adhesion GPCRs is probably true for PKDs, PKD1. PKD1 does have a gain domain, that is for sure, which is cleaved for sure. And I don't think that it is going to separate upon cleavage. And we can say that the extracellular region of PKD1 should be critical for downstream signaling. We probably don't know how extracellular region communicates with the TM region, and both the extracellular region and the TM region of PKD1 should be possible to be targeted for drug design. So basically, I think that in the PKD1, PKD2 field, there is the GPCR part, there is the channel part. Channels can be targeted by small molecules or antibodies that bind to them. GPCRs can be targeted by small molecules that bind to their transmembrane region. And now adhesion GPCRs, there's no one drug yet for adhesion GPCRs also, but I think PKD1 should be, my belief is that should be taking the signal from its large extracellular region, passing it to the transmembrane region and then to the channel. So the incoming of the signal, I believe, is from the extracellular region. And if there is any similarity between adhesion GPCRs and PKDs, the PKD extracellular region should also be important for PKD1 function and should be able to be targeted by antibodies for PKD1. So for that, and there is the gain domain there. For my acknowledgements, all of the work that has been done for adhesion GPCRs that we did is thanks to my lab, and I have many collaborators, which I forgot to list them here, sorry. But the PKD, I have briefly worked on PKD during my postdoc. It is a very large protein. I was trying to clone it, do mutations, polycystic kidney mutations on it to show whether they are, to test whether they were abolishing autoproteolysis or not. They are very difficult proteins to work with, I know that. And with that, thank you for your attention.
Thank you so much. That was a great presentation. So there are some questions in the chat, so I will start immediately from Alessandra Boleta. So great talk, thanks. Is there any GPCR that present with more than 7 transmembrane domains, or there are gain domains present in other types of receptors other than the GPCRs? So in the human genome, all of the gain domains, there are, I mean, GPCRs are 7-TM, that's what they are. Adhesion GPCRs have 7-TM. Gain domains, we looked at them where they exist. In humans, they exist in 33 adhesion GPCRs, and I believe 5 PKDs, PKD1s. There is no other gain domain anywhere else. But in earlier organisms, if you go to slime molds, for example, there is a gain domain that is connected to a protein, to a transmembrane region. I think it was a single TM. I don't know what it is. But wherever there is a PKD, I believe the C. elegans, honestly, I don't know the PKD field that much and I forgot. I think the earliest organism that there is a PKD1, is it C. elegans?
I think it is LAV1 in C. elegans, and it does have a 11 TM, I believe. I might be wrong, but I think so.
Thank you. I think that we have time for one more question, and the next one is from Robin Maser. So are there examples of adhesion GPCRs that are directly coupled to or directly modulated, that directly modulate the activity of an ion channel? Because you mentioned that they associate with G protein, but can they act directly with an ion channel?
So there is the latrophyllin 1 adhesion GPCR, which exists in humans all the way back to C. elegans, or maybe even earlier. In C. elegans, it is a LAT1. It's called LAT1. And what they have suggested, that they did find a genetic interaction. Sorry, did I say C. elegans? In Drosophila, it is called a CIRL, CIRL1. They have found a genetic interaction between CIRL1 and TRP channel. Basically, it is very similar. So they did reference the PKD1, PKD2 similarity, but there is in Drosophila, there is this CIRL1 and TRP channel similarity, where they are also saying that CIRL1, the adhesion GPCR, regulates mechanosensation. So TRP is the main channel that mediates mechanical, that takes the mechanical signal, but CIRL, latrophyllin, modulates it. It helps it. Without CIRL, the mechanosensation is messed up. So this is a genetic interaction they did show. They did not show a direct interaction.
Thank you so much. Your presentation was super popular. There are 3 more questions in the chat if you can answer, and I will give the word to Marcus to introduce the next speaker. Thank you.
Thank you.
Thank you very much for that talk. And so the last speaker of this session is Jonathan Jagger, who comes to us from the University of Tennessee Health Science Center. So John obtained his Ph.D. in ion channel physiology at the University of Sheffield in UK. And then he came to the US for postdoctoral training with Mark Nelson at the University of Vermont. And, so he is an established investigator and has been working, and his lab is at the, as I said, University of Tennessee. So he studies the function and the of ion channels in the endothelial cells. And he will be presenting, and the title of his talk is Blood Pressure Regulation by the Vascular Polycystin Proteins. John.
There we go. Can you hear me okay now? All right, great. Thanks for the kind introduction, and thanks especially for the invitation to speak in this symposium. This is my first time attending this symposium, but it certainly won't be my last. I'm pretty new to studying polycystin proteins. Seems like I'm having the same problem that Paul had, but I can't go through my slides. Nope. Oh, here we go. That was slow. All right, there we go. All right. So the focus of my lab is to investigate the regulation of arterial contractility and blood pressure by ion channels and calcium signals in arterial smooth muscle cells and endothelial cells. So this introductory slide shows you that there is both intercellular and intracellular signaling between these 2 different cell types. So if you look at a cross section through a small resistant-sized artery, we see that there are 3 primary cell layers. The tunica intima is essentially endothelial cells that line the lumen of all blood vessels. The tunica media is smooth muscle, and the arteries that regulate regional blood flow within organs and systemic blood pressure are called resistant-sized arteries, and those arteries have about 1 to 3 smooth muscle cells thick within the vascular wall. So the primary determinant or the primary calcium influx pathway within arterial smooth muscle cells is the voltage-dependent calcium channel, Cav1.2. And because that's a voltage-dependent calcium channel, membrane potential is a primary determinant of vascular contractility by controlling the amount of calcium that enters through the plasma membrane of smooth muscle cells. So an increase in intracellular calcium concentration stimulates vasoconstriction. Now, endothelial cells regulate the contractility of arterial smooth muscle cells in two primary ways. First of all, they are electrically coupled through gap junctions. So when endothelial cells hyperpolarize, this causes a hyperpolarization of arterial smooth muscle cells, and they relax, and therefore you get vasodilation and a decrease in blood pressure. Endothelial cells also generate a number of different diffusible substances, such as nitric oxide, which is another vasodilator, and that causes relaxation of arterial smooth muscle cells and vasorelaxation. So I was going to go through this. I thought Paul may actually introduce the structures that have been derived for PC2 and PC1, PC2 heterotetramers. So I'll skip these slides, but essentially what I'll like to point out here is that when studying physiological functions of polycystin proteins in smooth muscle and endothelial cells, we're really limited by the fact that there's few to no pharmacological tools available. And that's why what I'm going to present today is where we used genetic approaches in mice in order to study physiological functions of these proteins. So I'll skip this slide as well because Paul nicely presented this as well. So the first mouse model that we generated, we obtained the PKD2 flox mice from the Baltimore PKD core, and we bred that with what is the gold standard for an inducible, tamoxifen-inducible smooth muscle cell Cree model, which is a smooth muscle myosin heavy chain linked Cree. So in breeding those two mice, what we generated then was a tamoxifen-inducible smooth muscle cell Polycystin 2 knockout mouse. So of course we were interested primarily in does Polycystin 2 regulate blood pressure in these mice when it's expressed in arterial smooth muscle cells, and indeed that was the data that we obtained. When we treat these mice with tamoxifen, we see a decrease in both diastolic and systolic blood pressure, suggesting that Polycystin 2 in arterial smooth muscle cells functions to actually elevate blood pressure. Now, we did a lot of phenotype in these mice. We did echocardiography. We looked at their plasma hormones and electrolytes, urine electrolytes, and their kidney histology, and there was no change in any of these factors in the knockout mice. So in order to determine how Polycystin 2 in arterial smooth muscle cells regulates arterial contractility, what I'm showing you here is data where we have dissected out small resistant-sized hind limb arteries, which is a skeletal muscle artery preparation. And you can see that these arteries are very small, at about 150 microns in diameter. And systemic arteries and resistant-sized arteries generate what we call a myogenic response, where when you increase intravascular pressure, those arteries constrict in order to maintain blood flow within the organ. So shown in black here is data from our flox mice. If we increase intravascular pressure in arteries of the flox mice, we get a nice vasoconstriction, and the amount of vasoconstriction is shown here as myogenic tone. If we perform the same experiments in our smooth muscle Polycystin 2 knockout mice, we get a much smaller pressure-induced vasoconstriction, suggesting that intravascular pressure activates PC2 to induce vasoconstriction. So I limited the amount of electrophysiology that I'm going to show in this presentation. Here's one slide that I'm going to show you. And that's where if we patch onto arterial smooth muscle cells and induce a mechanosensitive response by causing cell swelling through an osmolarity change, in smooth muscle cells of the flox mouse model, we see a nice swelling-induced cation current that is essentially lost in the Polycystin 2 smooth muscle knockout cells. So just to summarize what we actually found in this study. So we looked at two different vascular beds. We looked at hind limb arteries, and we looked at mesenteric arteries, which are another important vascular bed for blood pressure control. And what we found is that Polycystin 2 channels are activated by two different stimuli in each vascular bed. I showed you in the previous slides that intravascular pressure activates PC2 channels in hind limb artery smooth muscle cells, Whereas in the mesenteric bed, we found that pressure does not activate PC2 channels, but the channels are activated by phenylephrine, which is an adrenoceptor agonist. And so it looks like adrenoceptors are coupled to PC2 channels in the mesenteric artery smooth muscle cells. Now, regardless of which bed we looked at and regardless of which stimulus activates PC2, what happens is we get an inward sodium current, which causes a membrane depolarization. And as I mentioned in the initial introduction slide, that depolarization activates voltage-dependent calcium channels on the plasma membrane, and this stimulates constriction and elevates blood pressure. So having this mouse model available to us, we wanted to know if targeting PC2 in arterial smooth muscle cells could potentially be an antihypertensive therapy. So we induced hypertension using this model, which is an angiotensin II induced model. So we implant mini pumps, which release angiotensin II into the mice. And within about 3 days, what we see is an elevation in blood pressure. And this is in black here is data from the floxed mice. If we do the same experiments in our Polycystin II knockout mice, we see attenuation of that blood pressure elevation, suggesting that PC2 could potentially be a antihypertensive therapy if it was targeted specifically in arterial smooth muscle cells. Now, one hallmark of hypertension is an increase in wall thickness. And so we did cross-sectional histology here of mesenteric arteries from our floxed and our knockout mice. And you can see there's an increase in wall thickness here in the floxed mouse model. But that wall thickness increase is attenuated in our PC2 smooth muscle knockout mice. And that probably is because of the attenuated increase in blood pressure here, because it's known that the increase in blood pressure is one factor that increases wall thickness in these mouse models. So just to summarize what we found, obviously I can't show you all the data here, I don't simply have time, but what we found is that hypertension induces a transcriptional elevation in PC2 expression in arterial smooth muscle cells. Those channels traffic to the plasma membranes. There's an increase in PC2 channels at the surface. And therefore, when those channels are activated, there are more of them, a larger current is generated, and that ultimately gives you a larger vasoconstriction, which contributes to the blood pressure elevation in those hypertensive mice. So moving on to our next study, what we noticed when we ran Western blots of arteries from our knockout mice is that we didn't just lose the amount of the predicted molecular weight band for Polycystin II. We also almost completely lost a band of a larger protein, which is almost 150 kilodaltons. And this just shows you here that this large band is almost completely lost in arteries of our Polycystin II knockout mice, suggesting that PC2 may exist as 2 different molecular weight proteins in arterial smooth muscle cells. So what we did was we ran gels of arterial lysate, and then we cut the gel at 2 different points and then sent those proteins off for mass spec to see whether or not this band actually is Polycystin II. And we did obtain identified peptide sequences for Polycystin II in both of these regions, which gave us some confidence that this band up here is actually Polycystin II. Now, a molecular shift of that magnitude suggests that Polycystin II is post-translationally modified. And if it is, then it has to be post-translationally modified by either sumylation or ubiquitination. And again, these are Western blots from arteries of mice, from mesenteric arteries. So if we perform immunoprecipitation, where we pull down protein using the PC2 antibody and then run that protein on Western blots. We reprobe the blots for SUMO1, SUMO2-3, which cannot be differentiated by antibodies, and ubiquitin, and clearly we get a similar size band up here, almost at 150 kilodaltons if we probe for SUMO1. So this gave us some faith that Polycystin II may be sumoylated by SUMO1 proteins. We did a variety of different things to confirm that. One was immunofluorescence resonance energy transfer, where you use 2 different fluorophores to 2 different proteins. If 2 fluorophores come within very close spatial proximity, in this case, 6 nanometers, you get a fluorescence signal. And you can see we have a FRET signal here that is at the plasma membrane. So this is the reaction that takes place when a protein is sumoylated. What we did was we purchased GST tagged polycystin 2, which has been purified. And then we put that GST tagged polycystin 2 into a reaction tube with all of the other components required for sumoylation. And then we ran those proteins on a Western and probed for GST. Now, this is the predicted molecular weight for polycystin 2. And what's obvious here is we have 3 other proteins which appear to look as if they are sumoylated by 1, 2, and 3 different SUMO1 proteins. So just to summarize this paper, our data suggested that in arterial smooth muscle cells, we have both non-sumoylated polycystin 2, and then we have SUMO1 modified polycystin 2. And although I didn't show you all of the results, our data showed that intravascular pressure actually activates the SUMO1 modified PC2. And that leads to membrane depolarization. And as I already showed, that activates voltage-dependent calcium channels, leading to vasoconstriction. We also found that this SUMO1 modified protein actually internalizes and recycles back to the plasma membrane if arteries are maintained at physiological intravascular pressure. And the reason that they recycle back to the plasma membrane is that the high calcium generated by intravascular pressure inhibits those proteins from being shuttled into a lysosomal degradation pathway. So if pressure falls or if calcium is decreased, these SUMO1 modified proteins are lysosomally degraded. From a physiological perspective, our data suggested that pressure actually controls the amount of SUMO1 modified PC2 at the plasma membrane to regulate vascular contractility and blood pressure. So we then moved on and made a tamoxifen-inducible endothelial cell-specific PC2 knockout mouse model. And again, we're using here the PKD2 Flox mouse from the Baltimore PKD core. And the gold standard for tamoxifen-inducible endothelial cell CRE is this Canherin 5 mouse, which we bred with the PKD2 Flox mouse to obtain PKD2 endothelial cell knockout mice. And here I'm showing you immunofluorescence, where PC2 is labeled in red. This is immunofluorescence of on-fast endothelial cells in small resistant-sized arteries. Nice labeling of PC2 in red here, which is lost in our knockout mouse model. Now, contrasting to what I showed you in the smooth muscle cell knockout mouse model, when we knock out PC2 in endothelial cells, what we see is an increase in both diastolic and systolic blood pressure. We did measurements for cardiac function, and we did Kinsey histology, and those were both similar to the Flox mouse model. Let me just point out that all of these measurements are done about 7 to 10 days after the last tamoxifen injection. So when we knock out polycystin 2 in our mouse models, we study them very quickly after knockout takes place. So there's not much time here for compensation to occur in the cells. So before I tell you how polycystin 2 in endothelial cells regulates arterial contractility, let me just tell you about the next study, which we published this year, where we made a polycystin 1 endothelial cell knockout mouse using the same Canherin 5 CRE model. And again, we have polycystin 1 labeling here in red, and that's lost in endothelial cells of resistant-sized arteries in our PKD1 endothelial cell knockout mouse model. So what we found is that knocking out either polycystin 1 or polycystin 2 attenuates flow-mediated vasodilation, which is a mechanosensitive stimulus in arteries that is known to cause vasodilation. So here we have increasing levels of shear stress, increasing flow, which causes vasodilation in our PC1 Flox mouse model. And there's clear attenuation of that flow-mediated vasodilation here in our endothelial cell-specific PC1 knockout mouse. And the blue line here is the PKD1 sensitive data. As I already mentioned, similar to what we see in the endothelial cell PC2 mouse knockout model, when we knock out PC1 in endothelial cells, we see an increase in blood pressure. So phenotypically, the data are very similar regardless of whether we knock out PC1 or PC2 in endothelial cells. So we made a double knockout just to make sure that that is the case. Were the proteins acting in series or were they acting in parallel? So here's our double flocked mouse model. Here's our double knockout mouse model. We have nice flow-mediated vasodilation in the flocked model, which is attenuated in the double knockout model, and it is attenuated the same as in the single knockout mouse model. So we phenotypically, it's the same as knocking out each one of these proteins. And here's the mean data for attenuation of flow-mediated vasodilation. Now, acetylcholine is the prototypical endothelial-dependent vasodilator ligand. It works through binding to muscarinic receptors on endothelial cells, and we do not see any change in acetylcholine-induced vasodilation in any of these endothelial cell-specific polycystin knockouts. So we have a stimulus-specific effect on flow-mediated dilation. So because we have evidence, thank you. So because we have evidence that polycystin 1 and polycystin 2 generate the same phenotype, we wanted to see whether or not PC1 and PC2 form a complex at the plasma membrane of endothelial cells. So here we're using a single molecule localization microscopy, which is a super resolution microscopy method, which has a resolution of about 20 nanometers. So here we have PC1 clusters in green. This is in the plasma membrane of endothelial cells. We have PC2 clusters in red, and then yellow is where those clusters merge. And you can see that we lose most of those clusters in our knockout mouse model. All of the clusters that you see here is non-specific labeling by the antibodies, which is normal for this kind of method, that you would see some non-specific labeling. Now, if we come over here, these are the clusters for PC1, these are the clusters for PC2, and this is where the overlap. Now, what's interesting here is when we knock out PC1, of course, we lose most of the clusters for PC1, but we also lose most of the clusters for PC2 at the plasma membrane. And we see exactly the same if we do the same experiments in our PC2 knockout mice, endothelial cells. So it looks as if PC1 and PC2 are both required in order for a PC1, PC2 complex to traffic to the plasma membrane. We do not see a decrease in the expression of the other protein when we knock out one of the polycystins. We only see a decrease in the amount of protein that is seen at the plasma membrane. So the distance between PC1 and PC2 is very short, only about 100 nanometers or so, suggesting that they do co-localize. And that increases a lot in our knockout, suggesting that this again is non-specific labeling. And flow does not alter the distance between PC1 or PC2 or the cluster overlap between PC1 and PC2. All right, so another electrophysiology slide here. So here we're measuring sodium current in endothelial cells, and we are measuring attenuation of flow-activated sodium current in endothelial cells when we knock out PC1 or PC2. Now, PC1 and PC2 are known to couple through multiple different regions or domains, one of which is the coiled domain on the C-terminus of each protein. And so we made peptides corresponding to the coiled domain on either PC1 or PC2, and we introduced those peptides, one of them through the patch clamp pipette to the interior of endothelial cells. And when we do that, we also see attenuation of flow-mediated sodium current in endothelial cells. So in black here is the control data, and in purple and orange here is when we introduce one or other of the coiled domain peptides. I'm almost done. I just have a summary slide now. Oh no, I have one more data slide. Let me just tell you about this too. So what is the mechanism by which PC1 and PC2 cause vasodilation? So we use blockers to endothelial nitric oxide synthase, to intermediate conductance calcium activated potassium channels, and small conductance calcium activated potassium channels. And we get far less attenuation of flow-mediated vasodilation by those blockers in the knockout mice, suggesting that PC1 and PC2 activate those proteins to cause vasodilation. And we also see attenuation of phosphorylation of eNOS, a site that's known to activate the enzyme. So just to quickly summarize, pressure or adrenergic receptor agonists activate SUMO1 modified PC2 in arterial smooth muscle cells. to cause vasoconstriction, whereas in endothelial cells, intravascular flow activates what we believe to be a PC1, PC2 complex. And this leads to calcium influx. I didn't show you that data, which activates small conductance and intermediate conductance calcium activated potassium channels, and eNOS to cause vasodilation. And just quickly to recognize the guys in the lab that did this work, that would be Charles Mackay, Dennis Leo, Rakib Hassan, and Simon Bully. And thanks to all of our funders for this work, funding this work. So thank you. I'd be happy to take any questions.
Yeah, thank you so much, John. That was a super interesting talk. Yeah, so there are 3 questions in the chat. I'll just read them out. So the first question is from Chris. What do you make of the finding that sumo-related PC2 does not quite make it up to that 150 kilodalton size you see?
So I'm not sure what the question's asking. What do I make by not quite making it to 150?
Well, I think, and the way that I would interpret this, it seems slightly smaller, the sumo-related. Do you think sumo-relation is the entire story? I mean, let me rephrase that question, or maybe Chris can ask, or yeah, other post-translational modifications. Do you think sumo-relation is the entire story?
Yeah, I mean, we don't know. I mean, essentially, we found evidence that PC2 is sumoylated, and there are, so when proteins, in order for a protein to be sumoylated, it needs to have a sumo interaction motif or a consensus binding motif. There are also, most proteins are actually sumoylated through non-consensus binding motifs, so it's kind of hard to find out exactly where sumo binds to many proteins that are known to be sumoylated because there is no consensus motif in that case. But sumo does actually have 6 total consensus binding motifs or sumo interaction motifs. So there is certainly, there certainly are some binding sites there that could accept a sumo-1 moiety. When you run sumoylated proteins on a gel, sumoylation can interfere with the way that those proteins run as well. So when you try and calculate exactly how many sumo motifs might be on your protein, it can be made difficult by the fact that the protein size may run differently because of sumoylation. So I'm not quite sure what the question was asking in terms of what do I make by not making it to 150 kilodaltons. We never saw protein that actually made it to 150 kilodaltons. It was just below 150 kilodaltons. And based on the shift that we saw when we ran the GST tag PC2, our data suggested that it was triple sumoylated.
Yeah, I think if the question, I think you guys can probably go in the chat or email afterwards. And yeah, I also had a quick question. So you were showing regarding the pressure sensitivity of PC2, you showed that in 2 different systems, it was either, also that's on your summary slide, it was either phenylephrine or pressure. But so how do you interpret that in one case it's kind of pressure sensitive, in the other system it's not pressure sensitive, or in
Yeah, so the pressure sensor in arterial smooth muscle cells is still not known. So the myogenic response was described over 100 years ago by a British guy called William Bayliss, and yet 100 years later we don't actually know what the intravascular pressure sensor is in arterial smooth muscle cells. So we don't. If we did know what it was, then of course it would be easier for us to determine how that sensor then couples to PC2. But as we don't know what it is, it's kind of hard to know. So the mechanosensor in endothelial cells is actually a flow sensor. It's not a pressure sensor. And so it's shear stress which causes vasodilation through endothelial cells. And there are several different proteins that are proposed to be the flow sensor in endothelial cells. Piezo is one of them. And of course, we know piezo may signal through calcium signaling to multiple different proteins. And as Paul discussed, it's known that PC2 is a calcium activated ion channel. So, you know, if you're asking me to throw out a hypothesis here, perhaps piezo is a pressure sensor that causes calcium signaling that activates PC1, PC2. That's one possibility. Of course, there are many others, but just throwing one idea out there, that could be the case.
Okay, I don't know. Do we have time? Thank you. Do we have time for more questions? Do you, yes, yeah.
Marcus, I think we're kind of due for our 5-minute
Good.
break. So anyone who has a question, please put it in the chat to Jonathan, and let's take a 5-minute break from 11:20, which it is now on Eastern Standard Time, to 11:25. Is that okay? Okay, so 5 minutes and we'll come back with our next session, which is latest advances in pediatric PKD.
Thank you so much.
All right, thank you.